So I'm Dorothy Tuthill. I'm the outgoing president of the American Penstemon Society, and I want to welcome all of you to this meeting. I really do wish that we were meeting in person. That would be so much nicer, but this is what we could do this year. I have several people that I want to thank for making this meeting possible. First of all, Andi Wolfe, who set up the Zoom meeting for all of us. Hugh McMillan is our webmaster. I know he can't be here tonight, but he helped a lot on getting everything arranged. And Martha Dibley and Jenny Maffett are our newsletter editor and publisher. And they worked really hard to get the most recent newsletter out under a pretty short deadline so that we could have all this information out on time. I think you probably all know, because I sent out emails, that we did set this all up on pretty short notice when we realized that, you know, even our plan B for an alternative meeting to Mexico wasn't going to be possible this year. I'd like to introduce our current board members and hope that they'll shout out if I say their name so you can see who they are.
They'll have to themselves because I've muted everybody so we don't have any extraneous noise. So as your name is called, go ahead and hit the unmute button.
Okay. So Lupita Wessler is our treasurer and has been for several years.
Hello.
Hi, Lupita. Thank you. Beth Corbin is our membership chair.
Hi.
Thank you, Beth. We have 3 at-large board members. They are Bob Pennington, Cindy Reed. I haven't seen either of them on here. Are you here, either of you? And Andi Wolfe, who you've already met, I think. And Ellen Watrous has been our secretary for the last couple of years, filling in. We also have, so that's our elected board members. We have a number of appointed members, and I already mentioned Hugh and Martha and Ginny. Also Rick Wessler, who is our seed exchange chair. Is Rick going to say hi?
Oh, he's in the background with Lupita.
Yeah. Thank you, Rick. And Steve Love, who is our bulletin editor. And then, of course, I want to thank all of our members.
Do you see anything
this
here?
As well.
I got my photo up.
Go ahead, Dorothy.
Okay. Next on the agenda was to be the teaser for next year's meeting. That was which we are planning for Utah. Keep your fingers crossed, everyone. To be presented by Mike Stevens and Steve Love. So since neither of them are on here, we will skip over that and hopefully at least one of them will join us in a bit. So that brings us to the Penstemon, or the COVID Penstemon Gallery. And can I share my screen, Andi?
You can hear it on the phone. See, there's a mute here, but that's not, I'm not muted.
I'm muting Donita, sorry. Dorothy, let me put you as a co-host. Okay, you are a co-host now, so you should be able to share your screen.
Okay, so before I share my screen as an introduction, hopefully you read about this in the newsletter and my emails, I propose that people go out and photograph Penstemon, since we couldn't get together this year and go out all as a group and photograph Penstemons. Then we had a few submitters here.
Switch to phone audio.
Okay, can people see my screen now?
Well, because
I can see it, Dorothy.
Okay, great. So we had 4 people submit pictures for this. And Beth Corbin gets the award for photographing the most Penstemons so far this summer, or at least photographing them and sending them to me. Some of you may have submitted more. With 21, Mark Egger did 8, Randy Tatro sent me 1, and then I've put in a couple as well. So our The goal originally was to try and get all of the taxa of penstemon photographed this summer, if possible. This is a way to encourage you all to go outside and explore. Of course, we're nowhere near all of that, but you can still go out and photograph and send me pictures, and we'll see what we get later. So I'm going to, I don't have all that were submitted on here because I thought that would take a long time. Let's see. Okay. So if we were in person, we would shout out answers as you figured out what these are, and I would toss you prizes or whatever. But we're not going to be able to do that on Zoom.
The chat box. Everybody
Okay.
can type the answer in.
Okay, yeah, we can do that. So if you think you know what this is, go ahead and type in your answer. And we'll see if we can get these. So this first one is one I photographed this year, and I put it in because if you came to the meeting last summer, we saw this species in North Park, Colorado. And it looks even better this summer down there. And a close-up of the flowers. I don't know if I can see the chat box, Andy. So if you're seeing any responses, shout them out.
Did you click on the chat itself? Down below, there's like a little cartoon bubble speech thing. And if you click on that, it should open up the chat window.
No, for whatever reason, now that I'm sharing my screen, I'm not getting that.
Oh, okay. All right. Well, so far I'm the only one that's guessed. Do you want me to tell you what my answer was?
Oh, well, here, I just clicked on it by accident anyway. Is that what you guessed, Andy?
That's what I guessed, yes.
Yeah, okay. Yep. Good job.
Is that Cyathophorus?
Yep, it's Cyathophorus.
Okay.
Okay, this one from a garden in Laramie, although it's native around here as well. Any guesses? There's only one photo of this one.
Is it in Wyoming?
It is in Wyoming. Okay, well, this is Penstemon glaber, variety alpinus.
So somebody said alpinus, so that's close.
Oh, good, yeah, good.
Christina.
Here's another Wyoming one, then we'll move on from Wyoming after this one. We're getting any guesses?
Kind of looks.
So this is Penstemon radicosus.
Radicosus, okay.
I think, you can correct my identifications if you want to, Andy.
I would have guessed grassless, but that's not right, so.
Okay, this is the one Randy submitted that he just took a photograph of yesterday, apparently, 12,000 feet up in the Colorado mountains.
Oh, that could be holly eye.
Yeah, and there's a close-up picture. Yeah, Randy thought it was holly eye.
Yeah.
He said it was really cool up there because the alpine forget-me-not and the Gunnison seagull lily were both blooming also in addition to this.
That's beautiful plant.
This is one of Beth's from her garden. Guesses? This is Penstemon Tonii firecracker.
Oh, you're still on the holly eye slide.
Oh, I am.
Yeah.
Okay. Let's see what I can do here.
Actually, I'm seeing the advanced slides.
What are people seeing now?
A red flowered thing.
Okay, so this is the firecracker, Penstemon Tonii. Can you see the name that popped up? Okay.
There we go.
Okay, have we moved on to a purple one?
You're on Cyananthus.
Yeah, okay. And another one of Beth's. So this one is Cyananthus subspecies subglaber. And this is a pretty cool picture that Beth sent in. It's typically white flowered. Is anybody guessing, Andy?
Well, actually, it's hard to know because the screen on my computer is like one behind. So I think
No.
it's because my internet connection is unstable, so I can't help you with this.
Okay, so are people seeing a lavender flower? This
Sort of.
is
that
Yes.
Yes, for goddess.
This is, so what Beth wrote in her email to me is a lavender deustis or deustis. A
Oh,
single
wow.
lavender one in a population of the typical whites. So that's pretty cool. Okay, another one from Beth's garden. Neotericus. I have to say, I never even heard of this one before. That was fun.
Rare plant from northeastern California.
No. Yeah, I want to come see your garden in person someday, Beth. Okay, so Beth sent a lot more than that. I picked out my favorites. And here's some that Mark sent along. So Mark's up in, I think he lives in Seattle. Anyway, he's in Washington. So his are from the upper Northwest. And there's a close-up picture of this one as well. So this one's Penstemon erianthurus variety white teddy eye. Another lovely pinkish one. This is the same one. Gardneri variety Gardneri. This is the only photo of this one, Glandulosus variety chilenensis. This one, according to Mark's notes, he took right in Seattle in a natural area. Here's a close-up of it. Penstemon ovatus. So are these pictures making you all want to go out and take your own pictures now and send them? I hope. And we have a close-up of this one as well. Pruinosis. I'm not giving you much time to guess anymore. People gonna want to guess? Uh-oh. There we go. Subserratus.
Oh.
Okay, so that's all I have to share with you from this, but when you all send in your other pictures, we'll put them in a gallery somewhere that's accessible to everybody, and we'll have a really nice bunch of photos. Okay, so I'm going to stop sharing. Let's see, has Steve Love joined us yet? I don't see him.
I still have 6 Hugh test only deletes.
Steve Love, are you on here? Doesn't sound like it. So we'll have to get Mike and Steve to give us the teaser for the next year's meeting that they've been planning another time, I guess.
They did, I heard from Mike what the date would be. It's the same weekend that I have the Botany 2021 meeting, and so be able to attend because I have to go.
Bad.
So it's I think it's the weekend of July 15th or somewhere in that range.
Okay, that sounds right. Yeah, it was going to be in mid-July. I remember that.
I won't be able to attend, unfortunately.
Okay, well, anyway, you won't get the teaser for that today, apparently, but you will be hearing all about it in upcoming newsletters. So as soon as you get the date, be sure that you mark it on your calendar. It will be in Utah. If I knew my Utah geography better, I would be able to tell you more about where it is, but it'll be great. So the last thing I want to do before I turn this over for Andi to give her talk is to recognize the new board members. Thank all of you who voted for our board members. And some of these names will be familiar. So Andi Wolfe will be our new president. This is for a 3-year term. Randy Tatro, the vice president. Cindy Reed has agreed to be secretary for the upcoming term. Lupita Wessler has agreed to remain as treasurer. Thank you, Lupita, for the great job you've done. Beth Corbin has agreed to remain as membership chair. Thank you, too. Bob Pennington will remain as an at-large board member, and our new at-large board member is Michael Stevens, who I guess we've already determined isn't here. But I really want to thank all of these people who stand up and do these things. And I can't say it too many times, this society is run by volunteers. And it really is important that people stand up and do these things for us so that we can have meetings and we can have newsletters and we can have a really nice bulletin publication too. So I really want to thank everybody who currently serves on our board and all of the people who are remaining on the board and all of the new people joining the board. And I know most of you are muted, but I'll clap anyway. Thank you so much.
Everybody do that.
So I think we're ready to turn it over to you, Andi, and hear your talk.
Well, thank you, Dorothy. And I want to thank you for serving as president for 3 years. It's a tough job, especially in hard times. So thank you for all you've done and keeping
Well,
the
thanks. You're
society.
welcome.
So can everybody hear me okay? Just give me a visual, something like that, so I know you're hearing it. So I'm going to do a combination of a slideshow and a video, and I hope that it will actually work. I have to remember to hit use computer audio so you can actually hear the audio for the video. But let me go ahead and get my screen sharing started for the slide talk, the slide part. So let's just do that now. And somehow I'm on the last slide. Let me stop my screen share and actually get it set up so I'm on the first slide. That would be very helpful, I think. So let's go all the way to the beginning here. And now I can share my screen one more time. There we go. And put it in slideshow mode. There you go. Can everybody see that? Just give me a thumbs up if you, okay, I see nod, so I'm sure everybody's okay. So when I was on the board meeting, we had a Zoom call a couple months ago, and I was showing some work in progress for the society board members, and they were so excited about the visuals that I was putting together for this biogeography survey that we were doing, that they said, oh, you're going to be the speaker at the Zoom meeting. I said, oh, okay, I could do that. That's great. So I want to give you a research presentation. I don't think it's too jargony. I think you can probably follow along, but it gives you an overview of what the research my lab group has been doing and what I've been working on essentially for the past 30 years. So the title of the talk is An Evolutionary Overview of Penstemon from Pleistocene to Present. And so I've got this organized into several parts. I'll start with an overview of Penstemon. All of you pretty much know a lot about Penstemon, but just as kind of a general overview for people that might forget some of the details. And then I'm going to go into some of the phylogenetics work that we have been doing as a lab. And I will talk a little bit about what those implications are for the taxonomy of the genus. And then getting into the more interesting things for our current work is testing adaptive radiation theory and also looking at the biogeography. So just as a background, you all know that Penstemon's a beautiful genus, has a lot of species. Currently it's 280 to 300 species, somewhere in the range there, but at last count it's probably around 285, and it keeps going up every few years. A new species is described. And it's not just 285 species, there's a lot of subspecific varieties. And so altogether we have around 380 taxa currently circumscribed. Traditionally, the genus has been divided into 6 subgenera, including Dacenthera, the Cryptostemon, Penstemon, Haberanthus, Acanthera, and Dissectus. And that's what the American Penstemon Society has recognized. But the Flora of North America treatment that just came out a year ago or so by Craig Freeman actually reduced the number of subgenera to 2, subgenus Dacenthera and subgenus Penstemon. In the traditional circumscription, we have 12 sections and 23 subsections. So a lot of you are familiar with how to key out Penstemons. A lot of the characters that we rely on for the first part of the key is to look at the anther-dehiscence pattern. And so, the subgenera that have traditionally been described are characterized by certain characteristics in the anther morphology. So Dacenthera has these big fat woolly anthers here. Cryptostemon has the same kind of anther morphology essentially as subgenus Penstemon. Subgenus Acanthera has this opening at the connective, looks kind of saccate. You can put googly eyes on these things and it's really fun. And then subgenus Dissecti also has a saccate anther morphology. And then subgenus Haberanthus is the most recent one that was recognized, has an anther-dehiscence pattern from the distal end up toward the connective and sometimes all the way up to the connective. So the anther-dehiscence pattern is very useful in keying out the species, at least to get them into the right part of the key. I've always been interested in this question: is it of phylogenetic or evolutionary significance? And I'll try to touch on that as we get into the phylogeny. So the distribution of penstemon is solely in North America. It goes all the way from Alaska down to essentially northern Guatemala, the mountain range down there. And it goes from coast to coast, but it's not in the Canadian Shield, and that's where the ice was pretty thick during the glacial maximums that we had during the Pleistocene. The epicenter for diversity is right here in Utah with about 75 or more species, probably 78 or more. And then this is kind of a heat map to show the number of species in terms of diversity. It gets a little bit less in some of the surrounding states, and then it starts to really fall off as you get east of the Rocky Mountains, and then falls off even more as you get east of the Mississippi. So I live right here in Ohio, and we have 6 species in Ohio. So I'm handicapped compared to all of you that live out west. I have 6 lovely white variety species of penstemon. You guys are really fortunate. So more than half of the species are in montane habitats. Most of them occur in xeric conditions. Some are actually in more wet conditions, more mountain meadows or mesic meadows. So just a typical habitat shot. This is Penstemon davidsonii. We have a lot of penstemons in Utah. So if you can make it to the meeting next year, please do. It's going to be fantastic. Some species actually occur growing straight out of rock. This is Penstemon petiolatus on limestone, and there's a close-up of that species. Some species, or in fact, I think most species that we collect are probably on road cuts like this. So this is Penstemon venustus. Some are actually in gravel, some are in deep sand. The one in deep sand there is Penstemon ammophilus. The one here in gravel is Penstemon atwoodii. And then some of them actually occur way out in the desert. So a lot of the species are in the Great Basin region, in the Intermountain region, and occur in pretty dry habitats. On the other hand, we have some things like Penstemon rydbergii and flavescens that occur in very wet meadows. And so that's quite a diverse range of habitats that they occur in. There's also a lot of morphological diversity, starting with the caudex, or the place where the stem meets the root. It can be woody or herbaceous, it can be branched, it can be a taproot. There's a lot of diversity there. Similar diversity in stems, leaves, the inflorescence, in flower morphology in the stamens, and also in the staminodes. So here's just a kind of a quick look at some of the diversity in habit. You know, they grow in all sorts of forms. The leaves are quite variable as you compare it across the genus. The most interesting one to me is the one right here. This is Penstemon dissectus, which only occurs in Georgia. Some of them have glaucous leaves, some of them have hairy leaves, some of them have entire leaves, serrate leaves, various configurations on that theme. And then the inflorescence morphology is quite diverse too. I have 2 favorites on this slide. One is Penstemon ambiguus, these beautiful, delicate looking inflorescences on these shrub forms, and Penstemon ammophilus, which has these fat inflorescence stems that are hollow inside, and they're just totally fascinating to me. Of course, the hummingbird species are also very beautiful. But when you start to do a comparison across the whole genus, you see this incredible diversity of floral types. And this is just straight on corolla pictures to show quite an amazing range of configurations, from nearly symmetrical in their radial settings. If you look at this one here, that's Penstemon ambiguus. This is Penstemon cerosensis. Some of them are really folded and convoluted. This is Penstemon personatus in that subgenus Cryptostemon. And then you have things that have hardly any upper petals and, you know, a big, well-developed lower petal system, and some that are just almost tubes. And so there's an incredible amount of diversity in just looking at the flower. Similar type of diversity in the length of the flower, all the way from the giants in Mexico. These are nearly 2 inches long, to these tiny little things that are less than maybe a ½ an inch or shorter long. And then the staminode diversity is also quite fascinating. So I have one of my more recent graduate students that joined the lab is actually going to be doing her dissertation on the significance of this difference in morphology in the staminode. So my lab has been working on penstemon my entire career, and primarily we've been looking at phylogenetics in the context of testing evolutionary theory. So did a collaboration years ago testing pollen presentation theory. Right now we're testing adaptive radiation theory. We've looked at hybridization and hybrid speciation. We've looked at conservation genetics, and we're also doing genomics. We actually have an entire genome for Penstemon petiolatus, and we're using that genome for work that my graduate students are doing to look at character evolution. So the first phylogenetic study that was published on penstemon was the work that I presented in 2006. And we were using ITS sequence data, which is a very short region of the nucleus, not very variable, but variable enough to get us some ideas. And then we used a couple of regions in the chloroplast genome. And so what I just want to show on this slide is that the origin of penstemon is right here in the ITS tree and right here in the chloroplast tree. And when you look at these lengths of the branches, this indicates the amount of change along those lineages. And from the start of penstemon, you can actually see that there's not a whole lot of change here along this backbone. This to me was intriguing and suggested that there was probably a phenomenon that we call a rapid evolutionary radiation. And I thought at the time that there probably was more to the story than just it's evolving very quickly. There has to be a reason that there's so many species of penstemon compared to, for example, Kekiella that has what, 7 species, and Coloni has 4 species, and Kyanophylla, its sister group, has only 2. And here we have penstemon with, you know, nearly 300 species. So to me, there was something really interesting going on. And so that's why we've continued to work on the genus. So current goals are listed here. We're looking at about 90% of the genus. We have a DNA bank that has more than 90% of the species of penstemon. And so we're doing a phylogenetic study with those DNAs that we've collected over the decades. We're examining the correlation of the phylogenetic patterns with the current taxonomy and with morphological trends. And we're using our phylogeny to test adaptive radiation theory. And also it serves a purpose for helping us to understand the biogeographic patterns in the genus. So I also have listed here at the bottom of the screen genomics, but I don't have enough time to even touch on that. So I'm not even going to mention it, just to let you know that we're doing genomics with the genus as well. So for our phylogenetic analysis, we've absolutely been very fortunate to get a lot of DNA sampling done through the kindness of people in the American Penstemon Society and my collaborators and colleagues that work in Les Mieles. So we've sampled every subgenus of the traditional taxonomy. We've sampled every section, every subsection, and we have about 94 to 96% of the species represented in the collection now. So the work that I'm going to present to you right now is what we've done with targeted amplicon sequencing for 243 species. And so we actually developed these gene regions from low coverage genomes that we just did shotgun sequencing and pulled in enough sequences to find where we had common genes across 6 different genomes. And so we sampled across the genus to try to find the genes that would actually be represented in all of those groups. And so we found 48 loci that looked like good candidates. Those were mostly nuclear genes and a few chloroplast genes, but when we actually sent them out for sequencing, only the nuclear loci worked for us. So we have 43 genes in this data set. We're also doing a technique called genotype by sequencing, and we have 265 species in that data set, but I'm not ready to present that yet. So for the 43 nuclear gene loci tree, we had more than 18,000 base pairs represented in this data set. And we used several really fancy ways of generating phylogenetic trees, all state of the art. I won't go into the details because it will just be jargon to you, but we have used the best methods that we have available to us for generating these trees. And so we also wanted to use that phylogeny to try to understand the taxonomy of penstemon, to see if these groups that we have been using as sections and subgenera, subsections, are actually natural groups. And what I mean by that, are they actually related in such a way that the members of that group are from common descent? So we wanted to see how they looked in a phylogenetic setting. So we developed, my former student, Paul Blischak, and I worked on this. We developed what we call a taxonomic bootstrap method. And basically that's just setting aside groups of taxa that are defined by their subgenera, by their sections, or by their subsections. And then we use those groups as a sampling pool. And we pull those out one at a time, just a sampling with replacement is what it's called. And we test those groups to see how they cluster, whether the sections come together, the subsections come together, or the subgenera come together. And so we actually do what's called a bootstrapping. And if the taxonomy is robust, the representation of that sampling would show up as high support, something above 70 bootstrap support in the tree. So I just wanted to set the stage so you know what I'm talking about when I show you those data. So here is the most current phylogenetic tree for 243 species. I'm sure you can see all the names on that screen, right? You can all read it and you all understand what that means. And I don't have to say anything else. Here we are, we've got the tree. That's nonsense because you can't read a thing and I won't tease you anymore. But you can see on the left-hand side of the screen, this is the phylogeny that is in the traditional presentation of a tree. But this side of the screen actually shows the branch lengths of those groups. And you can see, just like in the 2006 paper, these are really very closely related and evolving very quickly. These branch lengths are on the backbone are quite short. So this again is supporting this rapid diversification idea that we presented in 2006. What we have with 43 genes versus 1, essentially for the ITS, is we have much stronger relative nodal support throughout the tree. So we're pretty confident that our groupings are robust. So what I'm going to do is actually break this down into sections. So I'll start here at the earliest diverging lineages and work my way up to the most recently diverged lineage. And I'll just talk about those sections of the tree. So I'll start with the bottom section, section 1. This includes the subgenus Dacenthera, Cryptostemon and Sacanthera, and some other things. So where I have these red bars on the table here, so it shows the subgenus, section, subsection, and distribution. This is what will go into the publication that we submit for this study. So here is subgenus Dacenthera, and that has a 100% bootstrap, essentially, so that's a natural group. And whereas in the Flora North America treatment, Craig Freeman actually lumped Personatus into the Dacenthera based on my ITS tree from 2006. Our most recent study actually pulls it out, and it's now sister to the core group of penstemons, and that's with strong support. So Dacenthera, Cryptostemon, those are all good. The next diverging lineage here is Sacanthera. There's a kind of an anomaly. 2 of the taxa in that subgenus actually come out in a different placement than we would expect. So I'm not too worried about it, but here is the Sacanthera. And what is interesting and what's notable here is that Penstemon whippulanus is here, sisters to Serilatus, and the Gardnerii and Deusti are also a clade within the Sacanthera. So we're going to have to think about what that means. Sudans is here, and then we have Penstemon tracii. So the Deusti are not even monophyletic, but they are coming out here with the Sacanthera. So that's something to note. In the second section of the tree, we have subgenus Penstemon, and we have section Penstemon with subsections Proceri, Humulines, Penstemon, Tubiflori, and Multiflori. And the really interesting thing to note here is that the Humulines and Proceri actually are all jumbled up here in one clade. And this is something that David Keck had hypothesized back in the 1930s, that these are so closely related. Essentially, the only character that was separating the sections was whether the leaves were serrated or entire. So clearly that's not a robust character. But Diphyllus is also here from the Sacanthera, so that's kind of an interesting result. And then this is subsection Penstemon, but it also includes subsection Multiflori and Tubiflori. So that's with 100% bootstrap support, so clearly that's a natural group. The third section of that tree includes what we would call subsection or subgenus Dissecti and subgenus Penstemon, section Cespitosi, and the Cristati. And so the notable results here is that the Cespitosi needs to be expanded. We actually saw this pattern in the 2006 tree where Pinifolius comes out with the Linearioides group and Harborii came out with the rest of the Cespitosae. This is a very nice, well-supported group, very natural, but it doesn't really support two different subsections here. So this is probably all just section Cespitosae. Here's where Penstemon dissectus comes out. This was characterized as a subgenus, but given its placement in the tree, it's probably not valid to call it a subgenus any longer. The Cristati needs to be expanded. Here they have some of the things that were in this group actually come out in the Cristati. So we have Yampaiensis and Acalus and Larissifolius all come out here with the Cristati. The fourth section of that tree, we have Penstemon section Bacteriofoliae, Chameleon, Ceruli, and Gentianoides. And so one of the interesting results here is that several of the group from Gentianoides or Peltanthera subsection Centranthofoliae, if that's the APS taxonomy, those actually fall in here with the Cerulii. And Patens and Confusus also fall in here with the Ceruli. And then in the Bacteriofolius, which was a monotypic section, it actually is grouping with 100% bootstrap with two species that were in the fasciculus group down in Mexico. So this section will probably have to be expanded. And then the fifth section up in the tree includes what was considered subgenus Haberanthus and subgenus Penstemon, sections Ambigui, and Penstemon subsection Aeronari. And so what's interesting for this is that if you can see that I have GLs here and ELs here, it's clear that the traditional sections, Glavery and Elmigra, are not monophyletic. They're just all jumbled up in this one big clade that has very strong support. So clearly we're gonna have to rethink about that. Given the placement of this group in the tree toward the most derived area of the tree, I think we're probably going to have to do away with subgenus Haberanthus as well and reduce this to a section. Let's see if there's anything else I need to tell you about here. Oh yeah, I'm not sure about this result with ambiguous and alpha marginatus is coming as sister taxa. Even though I've gone back and I've re-looked at all the DNA accessions, I've looked at all the specimens, it's clear that there's a signal here, but we're not quite sure what to do with it. And then the most recently derived part of the tree includes some individuals from the Haberanthus and then Penstemon section Chameleon, Fasciculus, Gentianoides, and Peltanthera. And the notable results here is that there needs to be some expansion of the fasciculus. There's a couple of taxa from Haberanthus and also the one species from Penstemon section Chameleon that comes in here with the fasciculus group. And then we have some of the Haberanthus taxa need to be transferred into Gentianoides. There's the only thing that's really what you would expect to see without too much difficulty is this section here, which was Peltanthera, or also known as spectabules, and that includes the Petiolatus as part of that group. So if we think about the results of our phylogeny, we can kind of summarize some of the major patterns. Many of the groups hold together very well. There has to be a few additions and subtractions to actually pull those sections into more of an evolutionary schema. But the subgenera Dacenthera, Cryptostemon, Sacanthera, Penstemon are all probably okay, as long as we add a few taxa here and there. The sections, Cespitosae, Cristati, Ceruli, Haberanthus, Peltanthera, etc., those are mostly grouping together, but they may have to have some additions and transfers between sections. And some groups actually need a lot of work. Regarding the Anthrodehiscence, what I raised the question at the beginning, there seems to be one trend that I've noticed, especially in photographing as many species as I've photographed, is that there are taxa in section or subgenus Penstemon and all the way up to Dissecti that actually have the dehiscence initiating at the connective. And once you get past Dissecti in the tree, that switches over to the distal end. So there's a signal there that needs to be investigated, but I don't think it's something that we can hang taxonomy on. Let's get into how we can let the phylogenetic tree inform our taxonomy then. So this is the traditional taxonomy that you will see on the American Penstemon Society website. So we have the subgenera in black, we have the sections in blue, and the subsections listed there in kind of a purpley color. So that's what we started with in terms of using our phylogeny to try to understand the taxonomy. And this is what that looks like if we kind of superimpose the tree. So we have subgenus Dacenthera, Pleitstocene, Sacanthera, including some taxa from subgenus Penstemon. This is where subgenus Dissecti would show up in the middle of the tree. And then this part of the tree, which you can kind of call the crown clade of Penstemon, just has everything between Penstemon and Haberanthus totally jumbled up. So that, as a starting place, gives us a clue that we need to maybe revisit our taxonomy. So I told you about the taxonomic bootstrap method that we developed, where we actually sampled within sections, subsections, and subgenera, and reconstructed the evolutionary tree, the phylogenetic tree. And these numbers above the nodes here are significant in terms of if there's a star listed there, the topology matches the phylogeny, and it comes out consistently in that pattern. And where there's no star, that means that there's no relative nodal support to show that relationship in the tree. So what we're looking for here at the section level is to see that things that might be subsections of a section or things that would normally group together in the phylogeny actually do fall together. And the lack of support through here shows us that there's probably something that we have to look at very carefully in terms of the taxonomy. It makes a little bit more sense as we look at the subsections, because then we can actually sample more within a section by looking at those subsections. And so when we do that analysis, we see some of these groups actually do have strong support. So here's the node leading to the Cristati and Dissecti, showing their affinities, but the Cristati including some members of Aracopsis, so the Linearioides and the Yampyances and Caulices all go in with Cristati. This shows Penstemon, Tubiflora, and Multiflora are really strongly supported, but they're also nested here within Penstemon. And then the Cespitosi group has strong support. And then all the rest of that backbone shows patterns that we would expect. And the spectabules here with Petiolati and the Havardiani also come out together. So there are some areas in the taxonomy that agree quite well with the phylogenetic tree and others that need to be looked at. So what I would suggest is that we probably need some revision with subsections Humiles and Proceri. We need to expand Cespitosi to include some of those other taxa that consistently come in with them. Sacanthera needs to be expanded to include some of the subsections of Penstemon. Ambiguity needs to be looked at. Bacteropholi needs to be expanded. Chameleon needs to be looked at again. Fasciculus also needs some work. And Haberanthus needs to be downgraded to a section. And then we have to look at subsection Penstemon. So all of these things give us a starting point for looking at the circumscription of the genus. So here's where we are at this point. I propose that we should probably look at having 4 subgenera in Penstemon, and that would include Dacenthera, the Cryptostemon. They stay the same. Sacanthera needs to be expanded to include some groups of Penstemon, and Penstemon, subgenus Penstemon needs to be revised to include Haberanthus and Dissecti. And then we need to rearrange some of those sections and subsections. So that's where we are for the taxonomy at this point. So now I want to move into testing adaptive radiation theory, because this is what's really near and dear to my heart, because I've always been fascinated by why Penstemon is so diverse. Why are there so many species of Penstemon, and why are they so different from one another? And why do they occur in so many interesting habitats? So my most recent grant from National Science Foundation was specifically to test predictions of adaptive radiation theory using Penstemon as a model system. And so these are the predictions that have been proposed in the literature in terms of what constitutes an adaptive radiation. The first thing that you would look for would be an increase in morphological diversification and an increase in speciation rates from natural selection when organisms are presented with ecological opportunity. So that's a pretty clear pattern that you want to see. The second prediction would be that this kind of diversification would result from what's called a key innovation. And that's something in morphology, anatomy, physiology that really shows a pattern that once that area has been developed, that speciation just kind of takes off. So that's what we're calling an increased rate of diversification. And then the rates of diversification should be high initially and then should decline as ecological opportunity is lost or extinction rates increase. So these are the three primary patterns that we would expect to see. And so we've done quite a few different types of analyses to test adaptive radiation with penstemon. We had to start with a robust phylogenetic tree, and then we had to use that tree to establish a time frame. And so we developed what we call a time-calibrated tree using fossils to actually date the origin of Evolutionay and then the tribe, then the genus Penstemon. So we used a bunch of different packages. I won't bore you with these details, but again, these are the state-of-the-art methods of analyses that anyone uses when you're trying to use a phylogeny to test evolutionary hypotheses and ideas. So this is our time-calibrated tree. We actually pulled in groups of fossils in the Lamiales, the big order to which Plantagenaceae belongs. And so we used a couple of fossils from Bignoniaceae. We used 4 fossils from Plantagenaceae, and these were dated at divergence time between 2 of the Bignoniaceae at 28.4 million years, the one pair of taxa in Plantagenaceae diverged at 11.6 million years, and then another one diverged at 5.3 million years. So once we have this time frame from the fossil record, we can actually look at the DNA sequences for those taxa and put them in a tree with the taxa that we have collected. And so we actually used taxa from the Evolutionay and from Penstemon, and we did this rates analysis using this program. And so we were able to date the timing of Penstemon, the origin of Penstemon, to be around 3.7 to 3.4 million years ago. So the original genus origin was just before the Pleistocene. And then the crown clade, most of the rest of the taxa actually started to diversify about 2.5 million years ago, which is basically the time of the Pleistocene. So the majority of the diversification is correlated with the events of the Pleistocene. So this is the results of one of our analyses. This is kind of a heat map just to show you the pattern. So what we're looking for is the timing and the location of the net diversification for the tribe Evolutionay, including Penstemon. And so I have a couple of things here to point out. First of all, this is the tree. I don't know if you can see it on your screen, but there's a big red dot here. This is where Penstemon starts. And this is a heat map. So red means fast and blue means slow. So at the beginning of Penstemon, at the origin of Penstemon and its early diversification, the whole backbone up to about the midpoint is really diversifying very quickly. And then it's still diversifying pretty quickly in this section of the tree. And then as you start to get to the terminals clades, then the diversification rate slows down. And that means that it could be a pattern of extinction in there too, that we're not seeing the whole picture because we've lost taxa that we don't have access to anymore. But this pattern of a rapid rate of diversification and then a subsequent slowdown, that fits with an adaptive radiation prediction. You can see most of the net diversification rates here are actually in this middle zone. So this is extremely slow, this is extremely fast. Most of the taxa actually fall into this area of diversification, somewhere in the middle. This graph down here shows a net diversification rate for Evolutionay without Penstemon, and you can see that from, my mouse switches on me and I don't like that, from about 15 million years ago to present, the rate of diversification for the tribe without Penstemon is basically flatline. There's not an increase or it's not a decrease, it's just steady. When we look at the Evolutionay with Penstemon, you see that up until about 3.5 million years ago, it's pretty steady, and then boom, there goes Penstemon. And then when we look just at Penstemon, once it originated about 3.5 million years ago, most of the diversification has kind of been slowing down over time toward the present. So again, this is a strong indication that we're looking at an adaptive radiation. This is another way to look at that tree. It's easier to talk about characters when you're looking at a circle tree. So it's the same topology as the up and down tree that I showed you, but it's basically that red dot is here, and all of the clade diversifies around the circle up to this point up here. So I present it to you this way just so you understand what I'm going to be showing you in the next several slides. So what we're seeing is an increased rate early and a slowdown after groups diverge from one another. And that, again, is consistent with an adaptive radiation. So actually what that means, if you think about it, we have almost 300 species that most of them have evolved in the last 2.5 million years. That's kind of looking at one new species every 7,000 years. And I don't know if you know anything about the fossil literature, but that's really fast. Penstemons fast, and that's interesting. So this is a chart that has not a lot to do with penstemon, but I need to show you this so that you understand the pattern that I'm going to present to you in the next few slides. So this is a lineage through time plot, predictions about what would happen in a phylogenetic tree if you were looking at even rates of diversification through time, an early burst of diversification, and then just that's all you see, or a late burst of speciation or early extinction. And so these plots actually tell us something about how evolution is occurring. And so we wanted to actually look at these lineage through time plots with penstemon as we are looking at rates of diversification. So I'm going to show you several different character traits that we have looked at to see. We're looking for key innovations or a combination of key innovations to see what might explain why penstemon has evolved so quickly and diversified so fast. And so this is plotting lifespan onto the phylogenetic tree. And so a long lifespan is represented in those blue dots, and a short lifespan is represented in yellow. And when we look at the rate of diversification of those character traits on the tree, it turns out that the short lifespan is a higher rate of diversification than the long lifespan. And that occurs midway through the tree. And so it's not the early diverging lineages that have the short lifespan, it's the more recently diverging lineages that do. And we can kind of look at that with the lineage through time plot. That character for short lifespan is initiated about between 1.5 and 1 million years ago. And so it actually is increasing through time. So that's an interesting character trait. It's showing you that again. Okay. Another character trait that we looked at was the height of the plant. So most of you know that there's quite a range there. So the picture on the left is Penstemon acaulis, which is essentially, less than an inch or two tall. And then this is the most recently described species that my colleague Anna Gabriela Zacharia Correa and I described. That's Penstemon reedmoranii, which was probably misidentified in gardens as Penstemon eximius, but that's about 9 feet tall. So that's quite a range of height difference in the genus. So when we look at that, yeah, so that little white bar there, that's the height of Penstemon acaulis. Quite a change. So when we look at the rate of diversification on that character, it turns out that the tall character has a slightly higher rate of diversification than medium sized plants, but it's way higher than the short plants. And again, it kind of starts about 1.5 to 1 million years ago when that all starts to happen. So the tall one starts about the same time. We've also looked at corolla length. So corolla length varies from less than 10 millimeters to more than 30 millimeters or 50. And so we have a comparison here on this slide of Penstemon hartwigii that's at 50 millimeters or about 2.5 inches. And then Penstemon procerus, which is itty bitty. And it's the relative size is accurate here on this scale. So essentially Penstemon procerus is the size of the calyx of Penstemon hartwigii. And so that's the unit there. So we looked at corolla length, and it turns out that the medium corolla length actually is the highest rate of diversification. That starts again between 1.5 and 1 million years ago. And then I don't know why that's showing that twice. It's the same information, but that's okay. So the other character that I want to point out here, two more characters. One is the topography of the lower portion of the corolla. And so on the left here, we have a pleated morphology, so it's basically folded. And then we have a ridged morphology or a smooth morphology. Those are the three characters that we looked at. And when we looked at this rates analysis, it turns out that the pleated pattern actually has just been a steady rate, going on for the entire life cycle, or the time span for the evolution of penstemon. And then that actually is slightly higher, but the smooth is a fast competitor. So then the thread expansion is another character that we've looked at, whether it's from the upper part of the corolla, whether it's even on the top and the bottom, or whether the expansion is on the lower part of the corolla. And so when we look at that, it turns out that the lower corolla expansion has a much higher rate of diversification than the other morphologies. And again, that starts about the same time frame, between 1.5 and 1 million years old. Okay, so here's what we can conclude at this point from testing adaptive radiation. It's obvious that penstemon is a very young genus. The rate of diversification was even until about 1.5 to 1 million years ago, and then it took off. The rate of diversification has been rapid at first, and diversification of lineages have slowed through time, especially since the past 500,000 years. And then several traits have shown increased rates of diversification. And when we take all this information together, it's pretty clear that penstemon has undergone an adaptive radiation. We're not quite there yet to name all of the character traits. That's what my student Ben Stone and collaborator, former grad student Paul Blischak, are really concentrating their efforts on for the next few months. So then that brings us to the last topic. I hope you guys are okay with the length of the seminar, but it kind of gives you the whole picture. So I'm going to focus my information now on biogeography. And I'll start with Dick Straw's hypothesis back in the 1950s and 60s, where he hypothesized that, actually this was his 1966 paper, that Colonie originated in Northeast Asia, think Pinellianthus, and the tribe diversified via dispersal across Beringia and then south through the Americas. And this happened all in the early tertiary. And he also hypothesized that penstemon probably originated in the Colorado Plateau region of the Eastern Cordillera. He didn't give a time frame for that, but just based on its sister relationship to Cainophila, that's where he proposed the origin was. And so the goals for this part of the study were to assess the distribution and diversification based on a time-calibrated phylogeny. And we used the program called BioGeoBears, and we also manually plotted the distribution maps over time by downloading every species available in SciNet and getting the KML files and putting them in Google Earth so we could actually look at the distributions of the taxa. And so I have to put this in the context. We actually have estimated that penstemon originated before the Pleistocene started, just slightly before, but that the diversification starts really at the beginning of the Pleistocene and expands as we get into the Pleistocene. So I wanted to put that in the context of what the Pleistocene looked like. This is a chart from the World Meteorological Organization, and it basically shows where permanent glaciers are, where permafrost occurs, where there are ice shelves, where there's snow cover, and et cetera. And this is basically this part of North America during the glacial maximums is the only place that was not under snow cover and not frozen. So that's just something to keep in mind. This is what it kind of looks like. This is the extent of the glaciers at the last glacial maximum. So the ice occurred in this area, but you have to keep in mind that this area here was pretty much frozen or covered by snow during the maximum time of glaciation. And so we can look at the glacial maximum in the context of this red line. This is the zone of permafrost, and this area underneath here was probably snow covered most of the year, and then this area was basically snow-free. This is what North America looks like now. So going from here to here in the order of tens of thousands of years, that's a lot of change. And we know from all the fossil record, all the pollen records, that plants migrate north and south, depending on whether they're being pushed ahead of glaciers or following the retreat of glaciers. And so when you think about that, we always think about the last glacial maximum. That's the most recent ice age, which ended about 13 to 18,000 years ago. And you know, we always think that's the big event, that everything explains the distribution of plants from the Pleistocene is only the last glacial maximum. but the Pleistocene actually had 13 major glaciations, just like the last glacial maximum, and 9 minor glaciations, where maybe just the mountain ranges were under glaciers and the far north was under glaciation. But altogether, that's 21 events during 2.5 million years where glaciers covered a significant portion of North America. And the information for that comes from all of these geological sites. I'm not going to list them all, but these are the places in North America where they've actually studied the extent of glaciation. So keep that in mind. I'm going to actually want you to remember what the glacier extent was and where the permafrost was. We actually have been looking at the distribution of penstemons in the context of geographic zones. And these actually follow physiographic regions of North America that have been established by geologists. So I've actually broken that down slightly further just to kind of look at the distribution of penstemon. So if we were to look at zone 6 here, this is the Eastern Cordillera, basically the Rocky Mountains all the way into Canada. Zone 3 and 4 are basically the Cascade Sierra group and the coastal range that goes down into Baja. 5 is the Great Basin. 2 is the plateau regions. So this is the Pacific Northwest and the areas between the mountain ranges. And then this is a specific one up to Alaska. 8 is the lowlands. 9 is the Appalachian system. 10 is the coastal plain. 7 is the Great Plains, and 12 is the highlands of Mexico. So those are the regions where penstemon occur. So that's how we broke it up. And so we did the biogeobears. We did the manual plotting of the distributions using the KML data from SciNet. And so this is a tree that's generated in biogeobears. And all I want you to note from that is just the change of colors from the base of the clade to the tips of the clade. So when you see the same color, that means that they stay within their geographic zone. And then all of a sudden they switch to a new zone, and it basically just changes abruptly in some places here. So that's the pattern I just want you to think about. What the models that we tested show is a pattern of founder event speciation. So basically penstemon moves into a new area and diversification takes off. So that's a very interesting pattern. So you can have dispersal within a region followed by dispersal outside the region, and then the subsequent diversification. Again, this kind of fits the pattern for what we would expect with an adaptive radiation. Now I have to stop sharing this screen to show you the animation that I did. This is a 5-minute video. It has beautiful music to it, and I'll try to put it in context. There's a lot of printed information on there, but I'll just kind of remind you of what's happening in the Pleistocene as you see this. So let me stop the share on this, and then I have to start another screen share with iMovie. And I will just tell you that this is a work in progress. And the one caveat I have to tell you is that the distributions are only as good as the collection information that's recorded digitally in the herbariums. It doesn't include every herbarium record. And there are some species that aren't in this collection because they're rare or endangered or sensitive species. And we do have that data now because my student Mary Sakotaleva is working on another project, and she was able to get that, but not in time for me to plot it into this animation. The other thing that I want to state as a caveat is that we're looking at current distributions, not past distributions. So we don't know exactly what the past distributions look like, and we can estimate that individually doing what's called ecological niche modeling, but I haven't done that. So we'll just get a pattern look at this, and you'll see what I'm talking about. So I'm going to put this on full screen, and then I'll start the. Let me stop that. I just want to make sure I hit the computer audio thing. Okay, stop. Okay, sorry, I need to get out of that and get back into Zoom and make sure that you could hear the audio. So let me do that one more time. Share computer sound, there we go. And now iMovie, sorry about that. Okay, we'll start this again, here we go. Okay, so this starts at the beginning with the subgenus Dasyanthera. So I have the taxa listed on the left, and each distribution is 2 seconds in this video. During this event, there was one major glaciation and one minor glaciation. Now we're moving into a time frame 2.5 to 1.6 million years. During this time, there were 4 major glaciations, 2 minor glaciations, and not a lot of diversification in terms of what we're seeing here with the distributions. Now we're moving 1.6 to 1.4 million years ago, 2 minor glaciations. Again, not a whole lot of diversification going on at this stage. It's just starting. Most of the distribution is still in the Eastern Cordillera, but there are a couple of taxa that made their way over to the east. 1.4 to 1.3, things are picking up, there's no glaciation. And all of a sudden, things are expanding, contracting, going north, going south. So all those dots are the distributions in SciNet. 1.3 to 1.2 million years ago, wow, things are starting to take off. No glaciation during this time. So we see things going all the way up north to the Yukon, going over to the east, and filling in the places that were empty before in the west. Now 1.2 to 1.1 million years ago, there were 2 major glaciations. And again, the diversification kind of slows down a little bit. .1 to 1 million years ago, 1 minor glaciation, but diversification is going pretty well. We have 1 minor glaciation between 1 and 0.9 million years ago. So what you'll see is the glaciation, the diversifications in the south, and as it looks, it goes back north or it goes east and west. Okay, so now we're into 0.9 to 0.8 million years ago. There's 1 major glaciation here. Again, the number of species that we see is somewhat reduced. And then there's no glaciations 0.87 to 0.7 million years ago. And you'll start to see the Great Plains fill in this segment of time. .7 to 0.6 million years ago, we had 1 major glaciation event. And you'll see a lot of the diversification in this segment of time starts to occur more in the south. And also through the Great Plains. Okay, 0.6 to 0.5 million years ago, things are starting to slow down. And then 0.5 to 0.1 million years ago, 100 stop the share. I hope that was useful to you. I see Jenny. Okay, I'm going to go back to the slideshow now and finish that out. Let's see, where's my slideshow? There we go. Okay, so the geographic distribution was really significant in terms of understanding what was happening during that time frame. And we really had to have a time calibrated tree to be able to understand that. And again, the caveat is that the current distribution is not necessarily what was happening in the past, but it's probably within a range that is reasonable. So we can actually go back and revisit Dick Straw's hypothesis about the origin of penstemon, and he suggested the Colorado Plateau. Our data showed that's probably accurate. We're looking at the Eastern Cordillera, probably somewhere in the vicinity of the Colorado Plateau. And then diversification has happened primarily in the Western Mountain region, the Great Basin, the Intermountain region, Rocky Mountains, the Cascade Sierra, Nevada, and then down into the highlands of Mexico. And the chronological and phylogenetic sequences show a pattern that's strongly associated with the events during the Pleistocene. So I think that's quite interesting. Okay, and so what's happening during the glacial maximums and the retreat of the glaciers, the interglacial times, is that we have new habitats that open up. And so the mountains that were under ice, and snow actually become more temperate as the ice melts. And so we actually have new habitats opening up. The deserts and prairies, some of those regions may have been underwater. They dry out, they thaw. We end up with new habitats for plants to move into. And it's not just the plants and the ecosystems, for the soil profiles, et cetera, but we also have to look at what's happening with the pollinators. So whenever you have climate change, you can have extinction, or you can have diversification of the pollinators as well. And so as new ecological opportunities occur for the pollinators, that has an effect on penstemon. So we're actually inclined to think that the flower morphology, the pollination ecology, is actually very important for the diversification of penstemon. And we always kind of think about that in terms of the hummingbirds versus bee flowers, but actually most of the diversification is occurring in insect pollinated plants. And there's just a wide array of pollinators available to penstemon, and they exert strong selection pressure. So we're probably looking at the flower pollinator interaction as the key innovation for the diversification of penstemon. So just as a reminder, that's why, that's what we're seeing, is this incredible amount of diversity in the flowers. So I really thank you all for putting up with the virtual presentation. If you want to ask questions, we can do that. You can either type into the chat box, or I can let yourself unmute and you can ask questions. But I do have to make some acknowledgments here. National Science Foundation has been very generous in funding my research over my career, and most of my work has been on penstemon. And there's been just numerous collectors and collaborators that have helped provide material, including Mike Stevens, Dick Olmsted, Dave Tank, Paul Wilson, James Thompson, the Holmgrens, the Environmental Services, and then of course I've had just fantastic students over my career. So that's it for the slideshow, and we can certainly open it up for questions if you like. So do we have any questions? Oh, I see that, thank you. No questions?
I have a question, Andi.
Hey, Dorothy.
So the traditional 6 subgenera were defined by morphological features. Now with the kind of rearranging of some of the species inside of there, are there morphological traits that can be mapped onto those?
Yes, I believe so. And that's one of the things that you have to do if you're going to revise the taxonomies. You have to make the case that these things belong together. And so if we go to, for example, the Haberanthus group that have some of the gentianoides, or the ceruli that has some of the gentianoides in there, they all have glaucous entire leaves. They have character traits that seem to be relatively straightforward. So that's pretty clear. The case for the Pleitstocene, including pinifolias or including Harborii, you can look at their growth habit. That looks very similar. That's pretty easy to make the case for. Sacanthera, including the Deusti and the Gardneri. Well, one thing you can look at is where the anther to his pattern begins. And for those taxa, I know from photographing them that they start at the connective or toward the connective. So that seems to be a good character. There's commonalities in leaf serration for some of the Cereulati with like the Deusti. You know, there's characters I think we can find. Maybe my colleague Noel Holmgren would be kind enough to take that on, but I guess if I'm forced to do it, I'll have to make a list of characters, but maybe somebody will actually be kind enough to give me some help with that.
Good, that's reassuring, isn't it?
Yeah, you would hope that they would come together, absolutely. You don't want to be artificially classifying things just based on a phylogeny. But when the phylogeny is so well supported, I mean, that gives you an actual better basis for re-examining the morphology. And I don't know if all of you know, but there's a new nomenclature code out there. It's called the Philo Code, and it's based on phylogenetic analyses. And that just was published this year, just a few months ago. And so we have the International Code of Botanical Nomenclature, you know, that we all use, but there's now another possibility out there in terms of defining groups, and that's based on the evolutionary history of those groups. I see Hugh test only delete, but I think that, go. Let's see, I can't hear your microphone. Yeah, I can hear you barely. So can you turn it up a little bit?
I'll try to speak loudly. In the Colorado area, if you know, there was a lot of uranium sites that you use kind of ionizing radiation to suggest changes in your Penstemon.
You know, I'm very sorry, but I really can't hear you. Would it be possible for you to type the question in the chat box?
Yes, I'll do that.
Do that, and if somebody else has a question in the meantime, I'll take that. Any other questions? Okay. Okay, unmute yourself so I can hear the question.
What is the gorgeous Penstemon behind you? Is your screen safe?
So Dorothy played Name That Penstemon. Let's play Name That Penstemon. Who wants to guess? My students don't know this because I haven't used this in lab meeting yet. Every week in lab meeting, or most weeks in lab meeting, I play Name That Penstemon, and they go to the website and see which ones I've already put on the website. Any guesses? I will tell you it's in the Sacanthera. Is that a hint? It's in Utah, in the Wasatch Mountains. Okay, you all, in my lab group, this would be a cookie infraction. When somebody makes a mistake in my lab group, the next lab meeting, they have to bring cookies for the whole group. So next time I see you all, I expect some cookies. This is Penstemon leonardii, one of my very favorites. Okay, let's see. In Colorado, there's been a lot of uranium mined. Let's see if I can get to that. Uh-oh, I went back to full screen here. Pull up my Zoom, pull up the chat. Lots of uranium mined. Oh, that's a good question. Could radiation have an effect on diversification? In laboratory experiments, yes. I'm not sure anybody has actually looked at that in nature. One could make the case that that's a possibility, but you know, you could also look at extreme environments and see what kind of strong selection there would be on adapting to a weird environment like soil types or something. So in theory, radiation can cause mutations in the DNA, whether that's carried on in the germline or just in the non-germline. That's a good question, but it's an interesting idea, but I don't think anybody's ever really tested it. Okay, so thank you all for listening. I hope that was useful to you and that you have learned a little bit about the evolution and biogeography and all the stuff that we do in academia. We love to grow penstemons. I have quite a collection of penstemons in my own garden, but I really enjoy just going out and seeing them and photographing them and collecting them, and then being kind of mean to them and grinding them up and extracting their DNA, you know. So thank you so much. Dorothy, I'll turn it back to you.
All right. Thanks, Andi. That was great. Again, I can be clap for everybody because I'm unmuted. Yeah, thank you very much for that. Yeah, I guess I'm just ready to say goodbye to everybody and thank you all for joining us. It was a great pleasure to have everybody here. I'm really pleased so many people could come. Yeah, please don't hesitate to send me your photos so we can really put together a nice group of pictures. I noticed that Martha already sent me an email and asked if she could use some of those in upcoming newsletters. And so I'll check before I pass any of your photos on to her, but hopefully that would be fine with everyone. Yeah, we look forward to seeing everyone in person. I'm sorry, Andi, next year.
Yeah, actually what we'll have to do next year is on the Friday night session, we'll have to Zoom me in so I can actually be part of the business meeting.
Yeah, that would be great. Where is the meeting, the botany meeting next year?
Next year it's in Boise, actually.
Oh, well, so you could almost drive down and join us for an evening.
I could, it depends on when my paper, but the complicating factor, and I didn't mention this before, is I have a third meeting that's simultaneous.
Oh, darn it.
It is a science conference. It's my wood turning annual conference, and I don't finish there until Sunday evening.
No. Well, I'm sorry, but
yeah,
we'll do. Yeah. Yeah, so thank you everyone for coming, and we'll look forward to seeing you next year. And thanks again, Andi, for that great presentation.
Well, it's a pleasure. Bye, everyone. Thanks for joining us.